Okay, I'm going to go ahead and get started. So this week we're just doing a lecture. There's no quiz or exam today. So we're going to continue on with photosynthesis, which we started last week after the exam. And just to remind you of some of the things that we talked about, or a brief overview of what we talked about, we said that you could divide photosynthesis into two stages. And those two stages were the light reactions and the Calvin cycle. And in terms of, they both take place in the chloroplast, but the light reactions happen at the thylakoid membrane while the Calvin cycle happens in the stroma. And then just to relate it to these steps, during the light reactions you're going to do steps 1 and 2, so you're going to capture light energy and produce ATP and NADPH. And then the Calvin cycle is step 3, where you use the ATP and NADPH to make carbohydrates from carbon dioxide that the plant takes up from its environment. So last week what we did is we went through the details of the light reactions. And just to remind you, the light reactions, again, this is a big summary. You're going to capture light energy. We talked about how that happens. And then we talked about the pathway that produces ATP and NADPH. And then it also produces a byproduct of oxygen, which is not important later on in photosynthesis, but is important for aerobic respiration, which is our next topic. And the important products of light reactions are the ATP and the NADPH. And the reason that they're important is that the Calvin cycle depends on them. So we said that the light reactions are the photo part of photosynthesis, the Calvin cycle is the synthesis part, and the Calvin cycle is dependent on the ATP and NADPH made by the light reaction. So without the light reactions, the Calvin cycle will not take place either. So last week we focused on the reactions that occur during the light reactions. And now today we're going to start with talking about what happens during the Calvin cycle. Okay, so the first thing we'll see about the Calvin cycle is that it's a circular pathway. That's the reason it's called a cycle. And we'll see that carbon dioxide, so in terms of the Calvin cycle being the synthesis part of photosynthesis, this is where you're going to actually synthesize an organic molecule, which again is a carbon-containing molecule. And the source of carbon is going to be carbon dioxide that the plant takes up from the environment. So it's going to use carbon dioxide, and then it's also going to use the ATP and NADPH that you made during the light reactions to make a 3-carbon sugar called glyceraldehyde 3-phosphate. So the important product of the Calvin cycle, which you actually synthesize that the plant can use, is glyceraldehyde 3-phosphate. And then the plant can take that 3-carbon sugar and make other types of carbohydrates, including glucose, starch, cellulose, and in addition to that, other types of organic molecules. So it makes glyceraldehyde 3-phosphate first, and then it can make other organic molecules using that 3-carbon sugar. Okay, so when we talk about the Calvin cycle, we're going to break it down into 3 stages. And the 3 stages, the first stage is called the carbon fixation stage. And the reason that it's called that is because during this phase, you're going to take carbon dioxide, which is a gas, and attach it to attach it to an organic molecule. So you're basically fixing carbon dioxide in this. You're fixing it to something. So the first stage is the carbon fixation. The second stage is reduction and carbohydrate production. And this is the stage in which you actually produce glyceraldehyde 3-phosphate. So the important product of the Calvin cycle actually gets produced in the second stage of the Calvin cycle. So again, reduction and carbohydrate production. And the carbohydrate that we're referring to is glyceraldehyde 3-phosphate. Okay, and then the third stage is regeneration of a molecule called ribulose. bisphosphate, which from now on we'll just refer to or we'll abbreviate as RUBP. Okay. So I told you in the first stage, in the carbon fixation, you attach carbon dioxide to an organic molecule. Carbon dioxide is first fixed to ribulose bisphosphate in the first step of the Calvin cycle. And then as you progress all the way to the end of the cycle, you end up regenerating this molecule that you started with. And so that's why it's considered a cycle, because you regenerate that molecule at the end of the cycle. Okay, so each of these stages consists of multiple reactions. So it's not just one reaction. They consist of multiple reactions. But for this class, you just have to know what occurs during each of the stages. So you don't have to know the individual reactions. You have to know what occurs during the stages. Okay, so we're going to start, and I'll use the board first, and then I'll use the slide after we've discussed what happens during the cycle. So we're going to start, obviously, with the carbon fixation phase. And so the first step of this pathway is taking carbon dioxide and attaching it to ribulose bisphosphate. And ribulose bisphosphate is a 5-carbon molecule. That's why I'm just writing 5C in parentheses next to RUBP, because it's a 5-carbon molecule. Okay, so you're going to take carbon dioxide and attach it to ribulose bisphosphate. And this reaction is catalyzed by a very specific enzyme called ribisco. So ribisco is an enzyme, and it catalyzes the attachment of carbon dioxide to ribulose bisphosphate. For reasons that I'll explain later, we're going to talk about this pathway as 3 carbon dioxides enter the cycle. So if 3 carbon dioxides enter this cycle, each carbon dioxide gets attached to its own ribulose bisphosphate. So you have 3 carbon dioxides, 3 ribulose bisphosphates. Each carbon dioxide gets attached to its own ribulose bisphosphate. So if you attach carbon dioxide to this 5-carbon molecule, that's going to produce a 6-carbon molecule. And because you do that 3 times, you're going to, at this stage, have 3 6-carbon molecules. Okay, and the 6-carbon molecule that you produce at this step is very unstable, and immediately it splits apart into 2 3-carbon molecules that we'll just call 3PG, which stands for 3-phosphoglycerate, but it's a 3PG, 3-phosphoglycerate is a 3-carbon molecule. And so because you have 3 6-carbon molecules breaking apart, at the end of the stage, what you have are going to be 6 3-phosphoglycerates. Okay, so you start in this phase with carbon dioxide, you attach it to ribulose bisphosphate. If you have 3 carbon dioxides enter the cycle, you're going to end this phase with 6 3-phosphoglycerates. Okay, and again, 3-phosphoglycerate is a 3-carbon molecule. Okay, and then the next stage is the reduction and carbohydrate production. And so this stage is going to start with the product of the carbon fixation stage. So you're going to have 6 3-phosphoglycerates. So you have 6 3-phosphoglycerate molecules. And over a series of reactions, each 3-phosphoglycerate molecule is going to be converted into a glyceraldehyde 3-phosphate. So you're going to end this stage with 6 glyceraldehyde 3-phosphate molecules. And remember, this is the important product. This is the product that the plant can use to make organic molecules. Okay, so this stage is going to require energy in the form of ATP. So remember, we made ATP during the light reactions, and it's specifically required in this second stage. Also, given the name reduction, so you're reducing an organic molecule, so you need electrons for that, and you're going to use the NADPH that you created in the light reactions. And in this case, NADPH is going to be the electron donor that's used to reduce the organic molecule. So during this stage, you use both ATP and NADPH. Okay, so you produce six glyceraldehyde 3-phosphates in this step. And there's one of two things now that can happen with the glyceraldehyde 3-phosphate that you produce in this step. So what we're going to see is that when 3 carbon dioxides enter the cycle, 1 glyceraldehyde 3-phosphate can leave the cycle and be used to make organic molecules. So of the 6 that you produce, when 3 carbon dioxides enter, you can use one of those to actually do what you want to do, make organic molecules for the plant. Okay, what we're going to see then is that the other 5 continue on in the cycle. Okay, so 1 can leave and be used to make an organic molecule. And then the other 5 are going to continue on in the last phase or last stage of the Calvin cycle, which is the regeneration of ribulose bisphosphate. So this stage is going to start with the 5 glyceraldehyde 3-phosphates that are going to continue on in the cycle from the second stage. And glyceraldehyde 3-phosphate, remember we said, is a 3-carbon sugar. And you're going to use 5 glyceraldehyde 3-phosphates, and this is again not one reaction but several. You're going to use 5 glyceraldehyde 3-phosphates to regenerate 3 molecules of ribulose bisphosphate. And remember, ribulose bisphosphate is a 5-carbon molecule. And this stage also uses ATP that was made during the light reactions. Okay, so again, you regenerate this molecule at the end of the cycle that you started with at the beginning, and that's again why it's called a cycle, because you regenerate that molecule. Okay, but remember, the important product is produced here in the second stage, and the important product is glyceraldehyde 3-phosphate, but you actually have to use most of it to keep the cycle going. So most of the glyceraldehyde 3-phosphate that you make needs to go on in the cycle to regenerate the ribulose bisphosphate, and that keeps the cycle going. Okay, so those are the 3 stages of the Calvin cycle, and now I'll just use the slide. The different stages are not clearly marked on this slide, so I'll kind of explain as I go through it. But this also kind of puts it in a cycle rather than being linear like on the board. So the first stage we said is the carbon fixation stage. And you're going to have 3 carbon dioxide molecules. Each is going to be attached to ribulose bisphosphate. What they're not showing you in this cycle is the fact that you produce a 6-carbon molecule, and then that 6-carbon molecule breaks apart into 2 3-phosphoglycerates. So if you start with 3 molecules of carbon dioxide, at the end of the first stage, which is the carbon fixation stage, you're going to have 6 molecules of 3-phosphoglycerate. And then that's the starting material for the second stage, which is reduction and carbohydrate production, where you take 3-phosphoglycerate and transform it into glyceraldehyde 3-phosphate. So if you start with 6 3-phosphoglycerates, you're going to end this stage with 6 glyceraldehyde 3-phosphates. And you're going to use ATP and NADPH during this stage. So at the end of this stage, you have the 6 molecules of glyceraldehyde 3-phosphate. One of those molecules can be used to make organic molecules for the cell, so 1 can leave the cycle. The other 5 continue on in the cycle and are used to regenerate 3 molecules of ribulose bisphosphate, and then that stage also requires ATP that you made during the light reactions. Yep.
Does it always start with 3 molecules?
No, it's not. That's just something that we do it in 3s because you need at least 3 to make 1 that you can use to make an organic molecule. So this is just constantly happening. It's not like the plant is counting 1, 2, 3, okay, we can go. Carbon dioxide is just constantly entering the cycle. What that means is that in order to get one of these molecules that you can use to make other organic molecules, that's the minimum, yeah. So if it takes 3 carbon dioxides to make a 3-carbon sugar, which is glyceraldehyde 3-phosphate, how many carbon dioxides do you think have to enter the cycle if you want to produce glucose? Yes, 6. Yeah, so 6, yeah. So if you want to make a 6-carbon sugar like glucose, then you're going to have 6 carbon dioxides enter the cycle because you can use 2 glyceraldehyde 3-phosphate molecules to make a glucose molecule. Oh, you guys have other questions about the Calvin cycle? Okay. So the last thing that we're going to talk about in photosynthesis, or the last 2 things, first we're going to kind of talk about what happens in photorespiration, and then we're going to talk about the differences in terms of either not being able to minimize photorespiration or being able to minimize photorespiration. We're going to talk about the differences between C3 plants, plants that are called C4 plants, and then CAM plants. So we're going to start with talking about C3 plants. And as you can see from the slide there, most plant species on Earth are C3 plants, so they make up a majority of the plants that are found on Earth. And in C3 plants, initial carbon fixation, and when I say initial carbon fixation, I mean when is carbon dioxide first fixed to an organic molecule? And in C3 plants, initial carbon fixation happens through rubisco in the first step of the Calvin cycle. So in C3 plants, this is initial carbon fixation. The first step of the Calvin cycle takes carbon dioxide and attaches it to ribose bisphosphate. That produces a 6-carbon molecule, and this 6-carbon molecule immediately breaks apart into 2 3-carbon molecules. And because the product of initial carbon fixation is a 3-carbon molecule, that's where the name C3 comes from. So the C3 comes from the fact that the product of initial carbon fixation in these plants is a 3-carbon molecule. Okay, so I told you that rubisco is the enzyme that catalyzes this reaction. So what that means is rubisco's active site binds both carbon dioxide and ribulose bisphosphate. And it turns out that rubisco's active site can also, instead of binding carbon dioxide, can bind oxygen instead. Okay. And what happens if rubisco binds oxygen instead of carbon dioxide is it simply takes oxygen and attaches that to ribulose bisphosphate, but you're not, in that step, you're not going to be incorporating any new carbon into the plant because oxygen obviously doesn't contain any carbon. So normally for C3 plants, this is not a problem. Okay, so under normal conditions in which they're found, it's not a problem, but it can become a problem on hot, dry days. Okay, and the reason that it becomes a problem on hot and dry days is because plants have these openings called stomata. And stomata allow for gas exchange in the plant, so that's where carbon dioxide enters to be used in photosynthesis, and that's also where oxygen leaves, and that's the oxygen that's produced during photosynthesis. And then in addition to those 2 molecules, water can also escape through the stomata. So in terms of these plants, what happens in hot, dry conditions is they're going to close their stomata, and they're going to close their stomata to conserve water because they don't want to lose water under the hot and dry conditions. So they're closing the stomata to prevent water loss, but you're also going to prevent this gas exchange. So what that means is that on hot, dry days when you close these stomata, the plant is not taking up carbon dioxide and it's not releasing oxygen. So that means on the inside of the plant cells, your carbon dioxide levels are going to drop because you're using it in the Calvin cycle. So the concentrations of carbon dioxide are going to drop and the concentrations of oxygen are going to go up. Okay, so on hot, dry days when they close their stomata, closing the stomata results in conditions in which your carbon dioxide levels go down and your oxygen levels go up. Okay, now I told you that normally this isn't a problem because rubisco has a much higher affinity for carbon dioxide than it does oxygen. And for this class, you can just think of rubisco likes carbon dioxide better than it does oxygen. But when you have conditions where you start to raise the oxygen levels and lower the carbon dioxide levels, then that's going to favor this happening instead of this. Okay? And this here is the first step in photorespiration. And this is the only step of photorespiration that you need to know. But the first step of photorespiration is you take oxygen, Rubisco takes oxygen and attaches it to ribulose bisphosphate. And I previously told you that this incorporates no new carbon into the plant. So if photorespiration is taking place, that's going to limit the amount of carbohydrates or organic molecules that the plant can make. And then that is going to, because that's the source of food for the plant, that's going to limit plant growth. Okay? So what we're going to see is that in hot, dry environments, C3 plants have a disadvantage, and we're going to see that C4 and CAM plants have an advantage. And the reason that C4 and CAM plants are going to have an advantage is because they're able to minimize this process called photorespiration. Okay, so we're going to, we just talked about C3 plants, and on hot, dry days, photorespiration is going to take place. C4 and CAM plants have a pathway that minimizes that process, and so C4 and CAM plants have an advantage in hot, dry environments. Now, the other environment then would be cool and wet conditions, and in cool and wet conditions, C3 plants have an advantage over CAM and C4, and I'll explain why after we go through CAM and C4. Okay, so, but in cool, wet conditions, C3 plants have the advantage, but in hot and dry conditions, C4 and CAM plants have an advantage. Also, just in terms of the name, it's at the bottom of the slide there, but the reason that this is called photorespiration is because it happens in light, and it's a process that's going to consume oxygen, and then later on in this process you produce carbon dioxide, which is what happens in cellular respiration. In cellular respiration you consume oxygen and produce carbon dioxide. So because this process happens in light, consumes oxygen, and produces carbon dioxide, it's called photorespiration. Okay, and again, that's not something that the plant wants. It's going to limit the amount of organic molecules that the plant can make, and that's going to limit plant growth. Okay, do you guys have any questions so far? Yep.
If it uses the O2, we're still calling that the Calvin cycle.
No. So once it uses O2, that would be the first step in photorespiration, yeah.
So like the CO2 that gets produced, and does it get released, and that's why it's bad, or is the plant still good to
No, the reason that it's bad is because, remember, to make organic molecules you need a carbon source, and if you use oxygen instead of carbon dioxide, there's no carbon here.
Well, I'm just wondering, like, you said that it does get a CO2 like full, that it does have carbon dioxide in it. Like,
What?
photorespiration, like it produces carbon dioxide.
Mm-hmm.
Where does that carbon dioxide go? Does it get released by the plant, or
Mhm.
does it, like,
Yeah.
try to?
No, it would be released by the plant.
Okay.
Other questions? Yep.
How is it released if the stomata is closed? Or does that happen after it opens?
After it opens, yeah.
They can't use that to go ahead and make the...
It could, but the point is here is that when you have conditions where the oxygen levels are going up and your carbon dioxide levels are going down, that you're going to have this happening more often. So they're still happening at the same time. You're still doing this. You're just having this happen a little bit more than you did before. That's why you could still make organic molecules, but if you have this happening at the same time, that's going to decrease the amount because this is going to be happening less. So if this happens more, then this happens less. So it's not that the plant does one or the other, it's that they can both take place at the same time. And so the reason, again, that it's normally not, because you always have oxygen present, and the reason that it's under cool, wet conditions, it's not a problem, is because Rubisco has a much higher affinity for carbon dioxide, and because the plant is normally releasing oxygen, that's going to keep the oxygen levels in the plant low relative to the carbon dioxide levels, and because Rubisco has a much higher affinity for carbon dioxide, that means that it's going to favor this happening rather than that, which is good. That's what you want to happen, yeah. Other questions? Okay, so let's talk about what happens in C4 plants, and then we'll talk about CAM plants. And again, both of these plants are going to have a pathway that minimizes photorespiration taking place. And again, that's going to be the reason that they have an advantage in hot, dry conditions. Okay, so in C4 plants, they actually have, two photosynthetic cells. So they have mesophyll cells, which C3 plants have. And then they also have these cells called bundle sheath cells. Okay, and what we're going to see is that in C4 plants, initial carbon fixation does not happen in the first step of the Calvin cycle. And what these plants do is they separate that initial carbon fixation from the Calvin cycle. And how they separate it is into two different cell types. So what we're going to see is that initial carbon fixation, so the first time carbon dioxide is attached to an organic molecule, happens in the mesophyll cells. And it happens differently than previously discussed for C3 plants. So initial carbon fixation happens in the mesophyll cells, and then the Calvin cycle occurs in the bundle sheath cells. Okay, so remember for C3 plants do not separate initial carbon fixation from the Calvin cycle. In C3 plants, initial carbon fixation is the first step of the Calvin cycle, so there's no separation in C3 plants. In C4 plants, you separate these two processes. Okay, and so in C4 plants, what's going to happen, and this is showing you here the mesophyll cell in the picture, and then this is the bundle sheath cell. So we said this is showing you initial carbon fixation, and then the Calvin cycle happens in the bundle sheath cell. And in C4 plants, what happens is carbon dioxide enters the mesophyll cell and is attached to a 3-carbon molecule called phosphoenol pyruvate, which we'll just abbreviate PEP, unless you want to remember phosphoenol pyruvate. So you're going to take carbon dioxide and attach it to the phosphoenol pyruvate, and this is going to produce a 4-carbon molecule. And so the product of initial carbon fixation in these plants is a 4-carbon molecule. That's why they're called C4. Okay. And then this first step and these initial carbon fixation in these plants is performed by PEP carboxylase. So PEP carboxylase is the enzyme that attaches carbon dioxide to phosphoenol pyruvate. So this would be initial carbon fixation in C4 plants. The next step you don't need to remember, but what happens is you produce this 4-carbon molecule. It gets transformed into a different kind of 4-carbon molecule. But then after that, what happens is the 4-carbon molecule gets transported from the mesophyll cell into the bundle sheath cell. And then what happens is once it's in the bundle sheath cell, the 4-carbon molecule breaks apart and gives off carbon dioxide. So it breaks apart into carbon dioxide and pyruvate. And the carbon dioxide that's released in the bundle sheath cell then enters the Calvin cycle where it binds to ribisco and it's fixed to ribulose bisphosphate. Okay, so basically you've set up a system where this cell here is pumping, basically pumping carbon dioxide into this cell. And that's going to keep the carbon dioxide levels high in that cell, and this is going to minimize photorespiration. Okay, so in these plants, you've separated initial carbon fixation from the Calvin cycle into 2 cell types. Okay, and this pathway is going to minimize photorespiration. And then what you can see happens is the pyruvate that you produce when the 4-carbon molecule breaks down is transported back into the mesophyll cell and used to regenerate PEP, and that step requires more ATP. Okay, so this pathway minimizes photorespiration, which is why they have an advantage in hot, dry environments. But this pathway, in order to incorporate new carbon, uses more ATP compared to C3 plants. So in cool, wet conditions, that's why C3 plants have the advantage, because it takes them less ATP. They don't require as much energy in order to fix carbon as C4 plants do. But that extra energy that is required in C4 plants is going to minimize photorespiration, and that's going to give them an advantage in hot, dry environments. Okay, so that's what happens in C4 plants. And then CAM plants do something similar. So CAM plants also separate initial carbon fixation from the Calvin cycle. But rather than separating the two processes into two different cell types, a CAM plant separates them in time. So initial carbon fixation happens at night, and then the Calvin cycle happens during the day. So what these plants do is at night, when it's cool out, they'll have their stomata open and they'll take in carbon dioxide and produce a 4-carbon molecule. And then during the day, to conserve water, when it's hot out, they'll close their stomata, but then the 4-carbon molecule that build up at night is going to break down and give off carbon dioxide that enters the Calvin cycle. So you can see that this plant separates initial carbon fixation from the Calvin cycle in time. And again, initial carbon fixation happens at night, and then the Calvin cycle happens during the day. And that pathway also minimizes photorespiration in CAM plants. Okay. So when you compare the two, C4 to CAM, you can look at these two drawings at the bottom here. So this is supposed to be initial carbon fixation in both the plants. And then this is the Calvin cycle. And you can see that both of these types of plants separate those two things. They separate initial carbon fixation from the Calvin cycle. In C4 plants, the two processes are separated into two different cell types. So they're separated spatially. And then in CAM plants, they're separated in time, so they're separated temporally. Okay, if you were to do a drawing of a C3 plant next to those two, you would just have one cell, and it would be a mesophyll cell. And in C3 plants, carbon dioxide comes into the plant and directly enters the Calvin cycle. So there is no separation of initial carbon fixation from the Calvin cycle. The carbon dioxide that enters a C3 plant is initially fixed in the Calvin cycle. So you just have the Calvin cycle taking place in the C3 plants, and they're in the mesophyll cells of C3 plants. Okay, do you guys have any questions about C3, C4, or CAM plants, or the reasons why the C4 CAM plants will have an advantage in hot, dry conditions, whereas the C3 plants will have an advantage in cool, wet conditions?
So the CAM plants usually grow better than the C4 plants.
No. Yep.
Does the plant for the stomata of the plant, does it always close as soon as it takes on all plants?
No, no, no. Only when it's hot and dry out. So again, it's closing its stomata because it's trying to conserve water. So anytime the plant's going to want to conserve water, that's when it's going to close its stomata. Other than that, it's going to keep them open. Okay, other questions? Okay, let's do some of the lecture questions for this topic. Okay, so the first question that I want to talk about is what are the three products of the light reactions, and which of those products are used in the Calvin cycle?
Oxygen, ATP, NADPH.
Okay, right. So the three products are oxygen, ATP, and NADPH. And which ones are important for the Calvin cycle? What's that? Right, NADPH and ATP. And so those are the important products. Oxygen is a waste product of photosynthesis, but other life forms on Earth depend on the oxygen produced in photosynthesis because it's used in aerobic respiration. Okay, number 13, I'm talking more about in terms of the products. What, in terms of the products, is the difference between non-cyclic electron flow and cyclic electron flow? So they're both light reactions, but non-cyclic electron flow produces three things, whereas cyclic electron flow only produces one thing.
Non-cyclic produces oxygen, ATP, and ATP.
Right.
Cyclic just produces ATP.
Right. So remember, non-cyclic is the form that produces the three products, ATP, NADPH, and oxygen, whereas cyclic electron flow only produces ATP. And cyclic electron flow is a way of making more ATP without having to make more NADPH. And the reason that you would want to do that is because the Calvin cycle uses more ATP than it does NADPH. Okay, number 19, what is rubisco? It's an enzyme, and what reaction does it catalyze? Right. It catalyzes the first step of the Calvin cycle, where you take carbon dioxide and you attach it to ribulose bisphosphate. So it catalyzes the reaction between carbon dioxide and ribulose bisphosphate. Okay, so this is what happens in the first step of the Calvin cycle. In contrast to that, what occurs during photorespiration? Right, so the first step of photorespiration is rubisco taking oxygen and attaching it to ribulose bisphosphate instead of carbon dioxide. Okay, so I told you last week, and this is going to be true for aerobic cellular respiration, that you should be able to, I'm not going to ask you to write it out from memory, but in a multiple choice question, you should be able to pick out the right equation for both photosynthesis and aerobic cellular respiration. So which one of these best represents what happens during photosynthesis? Right, C. So you're going to use carbon dioxide along with water and then light energy, and you're going to make, that's going to make glucose. You get the byproduct of oxygen and water. Okay. Let's talk about the roles. What's carbon dioxide's role in photosynthesis? No, that's not the role of carbon dioxide. What's that?
Doesn't like get pumped there to keep ATP going.
No. What's the whole goal of photosynthesis? Yeah, or to make their own food, which is in the form of organic molecules, right? So where do they get the carbon to do that? It comes from carbon dioxide. So carbon dioxide is the carbon source in photosynthesis to make carbohydrates and other organic molecules. Does anyone remember what water's role is? And specifically, its role is played in the light reactions. Right, it's the electron donor in the light reactions. And then obviously light energy is the energy source that's needed in photosynthesis in order to synthesize organic molecules. Okay, so 26, we kind of discussed this in a short answer type of question, but The Calvin cycle uses blank and blank, which are products of the light reactions of photosynthesis.
D.
Right, D, ATP and NADPH. And then 27, the Calvin cycle begins by the attachment of carbon dioxide to which of the following? Right, A, ribulose bisphosphate. Okay, so 30 is asking you to take the different phrases and to put them into one of three categories. So either it's a phrase that applies to the light reactions, or it applies to the Calvin cycle, or it applies to both. So the first phrase, which category would you put occurs in the stroma?
Calvin?
Right, Calvin cycle. So occurs in the stroma, applies to the Calvin cycle. Where would you put produces ATP? Light reactions, right. So produces ATP, goes into light reactions. How about oxygen as a byproduct? Right, light reactions. How about captures visible parts of the electromagnetic spectrum? That's light reactions. How about uses NADPH? It's Calvin cycle. How about occurs in chloroplasts? Not that category. Oh, last one, both. So both the light reactions and the Calvin cycle occur in chloroplasts. So occurs in chloroplasts would go in the both category. Which one uses ATP? Right, Calvin cycle. Which one produces NADPH? Right, light reactions. Which one uses water? Right, light reactions. Remember, it's the electron donor. And which one produces glyceraldehyde 3-phosphate? Right, that's the Calvin cycle. Okay, so the next question is 36. The next question that I want to do is 36. So among the different forms of electromagnetic radiation emitted by the sun, which form is absorbed by plants for photosynthesis? Right, C, visible light. So we talked about the electromagnetic spectrum, which is all the wavelengths of electromagnetic radiation. And the wavelengths that are important for photosynthesis are the wavelengths that are part of the visible light part of that spectrum. So 37, which of the following is mismatched with its location? Right, D. And what would be the correct match? Right, thylakoid membrane. So D, to make D correct, it would have to be ATP synthase, and we said that that's embedded in the thylakoid membrane. Okay, 39, during non-cyclic electron flow of the light reaction, which molecule is the final acceptor of high energy electrons? What's that?
C?
Yeah, C, NADP+. So C, NADP+. Remember, the electrons that water donates in non-cyclic electron flow ultimately end up in a molecule of NADPH. So NADP+ is the final electron acceptor in that pathway. Okay, 40, which of the following represents a reactant or input for the light reactions of photosynthesis? No. So carbon dioxide is a reactant for the Calvin cycle, but not for the light reactions.
E?
Right, E, water. Okay, so ATP, oxygen, and NADPH are all products. Carbon dioxide is a reactant, but it's a reactant for the Calvin cycle, not the light reactions. Okay. Did you guys see other questions that you want to go through from this topic? We're going to go through 47 after we go through aerobic respiration. So we'll come back to this. But do you have other questions that you guys want to go through? 34? Right. So 34, which of the following, so this is, Which of the following is not a product of the light reactions? Right, D, carbon dioxide. So energy intermediates is referring to ATP and NADPH. So those are the energy intermediates that are produced. Oxygen is a byproduct of the light reactions. And then obviously you know that ATP and NADPH, we just said, are produced during the light reactions. Other questions that you guys want to go through? What's wrong with my what?
Oh, okay. Is it because of the spacers?
No, it's the S and the R are flipped and absorbed.
Oh, no. Well, I'm looking at like carbon.
What?
Oxygen. Oh, well, like the blue lines and the red lines.
Oh, the blue lines I think refers to the fact that when you have, you know how Word will, if you hit A and then you type something and you hit return, it'll automatically put B there. I've taken that out, and for some reason in Word, when you take that out, when it corrects itself like that, if you take that out and add in your own B, it underlines it in blue. That's what your test would look like too, if it was open in a Word document. Okay, other questions though from the lecture questions? 27? So 27, the Calvin cycle, so 27, the Calvin cycle begins by the attachment of carbon dioxide to which of the following? Right, A, so that was A, ribulose bisphosphate. Yep. 15. Oh, okay, so this is confusing because I shouldn't have written protons. So protons are hydrogen ions. So if you have a hydrogen ion, so if you have a hydrogen atom and the hydrogen atom loses an electron to become a hydrogen ion, all you're really left with is a proton because hydrogen is one proton, one electron. And so, sorry, I should have changed that too. So what I'm asking in this is, in which direction are hydrogen ions pumped during photosynthesis? So do you know the answer to that one? No, so pumped is when they get moved against their electrochemical gradient. So this is going to create the electrochemical gradient that allows hydrogen ions to flow through ATP synthase. So in which direction, and I'm asking into what area are hydrogen ions pumped during photosynthesis? Right, the thylakoid lumen. So in photosynthesis during the light reactions, the hydrogen ions are pumped into the lumen. That concentrates them there. And then what happens is the hydrogen ions are going to flow down their electrochemical gradient from the lumen into the stroma through ATP synthase, and that powers ATP synthase. So initially they're pumped into the lumen, that concentrates them, and then they flow through ATP synthase, and it's that flow of hydrogen ions that powers ATP synthase. Other questions? Okay, so let's take a break, and then when we come back from the break, we're going to start in on cellular respiration and fermentation.